Experimental design, the null hypothesis, and how to control nuisance variables. This video will look at experimental design and how to lay out an experiment. This will only cover some of the basics, but we'll look at some serious questions. Key steps in designing your experiment. Experimental design is extremely important as it is the basis of a well-conducted experiment. If the experimental design is flawed, then the results will be flawed as well. Poor experimental design will cause the results to be flawed, which will make them unreliable. When unreliable results are used, any conclusions or ideas inferred from them will also be flawed. Experimental design begins with the variables. Consider your variables and how they are related. How can these variables be used to determine the results that are going to be gathered? Write a specific testable hypothesis. The lack of a testable hypothesis will result in an experiment that will not provide any usable information. The hypothesis must include all of the variables to be tested. Design experimental treatments to manipulate your independent variable. The independent variable must be identified and varied. This will give you conclusions that can be tested. It is possible to have more than one independent variable, but then it becomes difficult to determine just what is causing any results. Assign subjects to groups, either among groups or within groups. Within any experiment, you assign variables to be among groups, that is, between the various groups, or within groups. An example of this would be feeding fiber to various cattle. Let's say you have 5 levels of fiber, with 5 animals in each treatment. Among groups would compare the groups with 5 animals to each other groups of 5 animals. We would look at the level of fiber within each group. The other is within group. Each of the animals will be similar, but they will not be identical. With plants bred for food, the germplasm is often identical as they are descended from a small number of original seeds. With animals, even very distinctive purebreds, there will be considerable genetic variation between lines. This needs to be compensated for in the experiment. Sometimes we talk about nested designs. This would be something like dealing with nutrition of chicks. You would have 5 different hens, each laid 10 eggs, and then study the chicks. Since each group would have similar genetics, we would use a nested design. Plan how you measure your independent variable. With this part is the procedure for measurement as well as the accuracy. With different kinds of instruments, you can get variation in measurement. This needs to be accounted for. Hypothesis. The hypothesis is a potential answer to the question. Since we start out with the research question, we will then need to create a hypothesis. This would be a potential explanation for the research question. We often call this the alternative hypothesis. Alternative hypotheses therefore predict differences based on treatment. The concept is that you cannot prove that something did occur, but you can prove that something did not occur. With scientific research, we can always determine that something did not occur. Let's use the current subject of COVID vaccines. It is easily proven that a vaccine did not work. Therefore, we create the null hypothesis that predicts no difference based on treatment. We can run the experiment and determine whether there was some sort of resistance to the virus. Reject or fail to reject. We test the null hypothesis, and based on the results, we can reject or fail to reject the null hypothesis. Reject the null hypothesis indicates there were differences in our treatments. So if the test on the COVID vaccine appeared to provide resistance, they would reject the null hypothesis in favor of the alternative hypothesis. Fail to reject indicates there were no significant differences based on treatment. If the vaccine did not work, then we would fail to reject the null hypothesis. The next question we deal with is, do we accept? We do not accept one hypothesis over the other. In science, there is always room for more knowledge, which will change the way that we understand something. We find information that supports our conclusions. But if new information becomes available, then our conclusions may change. What if we did something wrong? What if there was something we did not take into account? What if? We all witnessed this with COVID. Every day there was new information, and some of it contradicted what was previously believed. With that, the concept changed. Today, what we know is very different than what was believed in the beginning. Control variables. A control variable, or scientific constant, is an experimental element which is constant and unchanged throughout the course of the investigation. Under laboratory conditions, the temperature can be held constant. Temperature will affect the outcome. Temperature can be a controlled variable when we are doing an experiment. Also, temperature could be one of the monitored variables. For example, if we wanted to compare the growth of E. coli at 20, 25, and 30 degrees Celsius, temperature would be the independent variable. Nuisance variables. Nuisance variables are those that can affect an experiment but are not believed to have an interaction with the variable to be tested. In a field experiment, plants are subject to sunlight. Sunlight is important for plant growth. Over the time of the experiment, cloud cover varies. Day length varies. Angle of the sun relative to the plant changes. Since all of the plants are subject to these, they are believed not to have an interaction with the tested variable. Replications. One of the major things we look at with an experiment is the number of replication or data points. It is necessary to create a proper layout so that all of the possible variables are accounted for. Set up a sufficient number of replications so that the variation can be determined. A general rule of thumb is there are a minimum of 3 replications. The number of units in a replication depends on the kind of experiment. For example, if we are looking at the percentage of seed infected, a sample size of 200 seeds with 3 replications would be indicated. If we are looking at calcium uptake in plants, we would have a sample size of 10 plants with 3 replications. The more observations you have, the more accurate you can measure variation. Going back to the COVID vaccine research, in phase 1 tests, which use healthy adult subjects, there are less than 100 subjects. This will test safety, dosage, and side effects. In phase 2 tests, from 100 to 300 subjects are used to test efficacy and side effects. Phase 3 tests use several hundred to a few thousand test subjects from the general population. This will test effectiveness and gather more data on safety and interaction with other conditions. After the phase 3 test, a material can be liberated for general use in the population. Again, constant studies are needed for more information about safety, effectiveness, and interactions. In this case, the sample size is several thousand subjects or more. The larger the study, the better the results. Layout. The layout is the general pattern that we use to create the study. Layout depends on the experiment. 3 basic designs that are often used in biological studies include the Latin square, a randomized block design, or a completely randomized design. Latin square is often used in field studies and controls 2 sources of nuisance variables such as east-west, north-south variation in the soil. Randomized block is often used under controlled conditions or under greenhouse conditions where one factor is of primary interest and there may be several nuisance factors. Completely randomized takes all of the replications and treatments and completely randomizes them in the same block. We will go over each of these in separate videos. Experimental design, therefore, is of extreme importance. Without proper experimental design, any result that we may obtain may be questioned.